For decades, the playbook for treating psychosis has been pretty much the same. But what if we've been going after the wrong target all along? Today, we're diving into a really different approach in mental health, one that zeroes in on a kind of hidden dimmer switch in the brain that could totally change how we treat these conditions.
So the big question here, the one driving all this research, is a really powerful one. Can we find a way to treat conditions like schizophrenia that not only works but also dodges all those debilitating side effects that have been a problem for patients for so long? For anyone in drug development, that's been the holy grail.
So what's the deal with our current treatments? Well, a lot of the antipsychotics out there work by directly blocking dopamine receptors in the brain, specifically the D2 receptor. Now that can help with psychosis, sure, but it's kind of like using a sledgehammer to fix a watch. It can lead to some really serious movement disorders, major weight gain, and other metabolic issues. But this new approach we're talking about, it's way more subtle. The idea is to gently modulate or fine-tune the dopamine system from the side, hopefully sidestepping all those nasty side effects.
So, how are we going to break this down? First, we'll look at why this is such a new hope for psychiatry. Then, we'll get into our main character, what exactly is TAAR1. After that, we'll see how it acts as a master regulator for the whole body, not just the brain. We'll follow the decades-long hunt for a drug to target it, and finally, we'll look at the clinical trial data and ask the big question, is this the breakthrough everyone's been waiting for?
All right, let's get to the star of the show. What exactly is this hidden controller that's got scientists so excited?
TAAR1. It stands for Trace Amine-Associated Receptor 1. It's a mouthful, I know. The easiest way to think about it is like a dimmer switch for the brain's most important circuits. It's a type of receptor, which is basically a lock on the surface of a cell. When the right key comes along and fits in the lock, it sends a signal. But here's the cool part. Instead of just flipping the dopamine system on or off, TAAR1 just gently dials the activity up or down.
Okay, so what are the natural keys that fit into this TAAR1 lock? They're called trace amines, and your body is actually making them right now as we speak. These are molecules with names like phenethylamine and tryptamine, and they're constantly interacting with these TAAR1 receptors, helping to keep your brain chemistry humming along in balance.
And this is where it gets really interesting, the genetics. It turns out the genes that give our bodies the instructions to build these TAAR receptors, they're all huddled together on chromosome 6, in the exact same neighborhood that massive genetic studies have already linked to schizophrenia and mood disorders. I mean, for a scientist, that's not just a clue. That's a giant flashing neon sign saying, look over here.
So a really crucial point to get here is that TAAR1 isn't just some bit player stuck in the brain. No, its influence is everywhere. It's acting like a master regulator for the entire body.
Just take a look at this diagram from the source paper. It really says it all. You can see TAAR1 isn't just in the brain. It's in the pancreas, the liver, the intestines, the immune cells. It's all over the place. And this widespread distribution is the secret to both its potential power as a medicine and its potentially cleaner side effect profile.
Okay, so let's zoom in on the brain for a second. In here, TAAR1 is all about that fine-tuning. We're not talking about big, dramatic changes, just those subtle adjustments that keep everything running smoothly.
So how does it do this fine-tuning? Well, it gets involved in the electrical properties of neurons, it helps regulate ion channels, it influences these critical signaling pathways, but the big one, the one you really need to remember, is that it adjusts the firing rates of the very same dopamine and serotonin neurons that are the targets of so many of our current psychiatric drugs.
Okay, but let's pan back out to the rest of the body, because this is just as important, especially when you think about those metabolic side effects of older drugs, things like weight gain and the risk of diabetes.
So in the pancreas, it helps kick up insulin secretion. In the liver, it can help bring down fasting glucose. It's also active in the gut and even the immune system, so just stop and think about that for a second. A drug that turns on TAAR1 might not just avoid causing those awful metabolic problems, it might actually help with them.
And that right there brings us to the absolute core idea. By activating TAAR1, you can cool down an overactive dopamine system without having to slam a roadblock down on that main D2 receptor. It's this elegant, backdoor approach, and that is the key to this entire story.
Okay, so how in the world did scientists get from this really cool biological idea to an actual pill somebody can take? Well, it was a hunt that took decades of really dedicated work.
The story really kicks off in the early 2000s when these receptors were first discovered. Then, in the 2010s, the hunt began for real. A drug company, Roche, did this massive screen, testing, get this, almost 800,000 different chemical compounds to see which ones would talk to TAAR1. That huge effort paved the way for another company, Synovion, to eventually take its own compound all the way into human trials.
But, drug development is never that easy. One of the biggest hurdles is just pharmacology. Because of our own unique genetics, you and I could take the exact same pill, the exact same dose, and end up with completely different amounts of the drug floating around in our bodies.
And this chart right here, it just lays the problem out perfectly. For one of the early drug candidates, they found that the gene for being a poor metabolizer, which means the drug hangs around in your body for a long time, is in 45% of people of African origin. 45%! But then you look at people of Asian origin, it's only 8%. And for Caucasians, less than 1%. This just goes to show you why a one-size-fits-all dose can be really dangerous and why finding the right compound is so tricky.
And that all leads us to the climax of our story today, the first major human trial of a drug designed specifically to turn on TAAR1. The compound is called SEP363856.
All right, let's meet the drug itself. SEP363856 is what's called an agonist, which just means it activates TAAR1. It also has some activity at a serotonin receptor. But here's the kicker, the part they really stress in the research. It's what it doesn't do. It does not block those D2 dopamine receptors in any significant way. It's a whole new ballgame.
So when the results of the first big trial came out, published in a major medical journal, they were pretty striking. Let's just walk through the numbers here. Patients having a schizophrenia episode started with a symptom score around 100. After 4 weeks, the folks on placebo actually got better by about 10 points, and that's pretty normal. You expect a placebo effect. But the group taking the new drug, they improved by more than 17 points. That difference was statistically significant, and it pointed to a clear, real benefit.
So, you can imagine the researchers who wrote the paper we're looking at were pretty excited. They basically concluded by saying that TAAR1 modulators could become a whole new class of drugs for managing a wide range of mental health conditions down the road.
And the potential here just explodes from this point. I mean, we're not just talking about schizophrenia anymore. This same mechanism is being looked at for psychosis in Parkinson's disease, for substance use disorders, and because of all those effects on the body we talked about, even for things like metabolic syndrome and obesity. It's an incredibly versatile target.
Now look, we have to be clear. This is just one study. A lot more research still needs to be done. But it leaves us with a really profound and hopeful question. After so many decades of relying on the same basic approach, are we standing on the brink of a totally new era in how we treat some of the most challenging conditions of the human brain? Only time is going to tell.